I'm Conor Loy. I'm the CEO and co-founder of Romix Biosciences. I'm currently an Ignite Fellow, 6 months through, so halfway through the program, and looking to launch this spring or summer. So at Romix, what we are interested in doing is helping doctors better diagnose and to ultimately treat patients with inflammatory diseases. Now, inflammation is really complex. It's interactions between the immune system, our solid organs, pathogens, and the environment. And unfortunately, we desperately are lacking tools to really measure this complex interactions. Because of this, patients are suffering and dying because they aren't getting diagnosed in a timely or accurate manner. And populations such as children or cancer patients are particularly vulnerable to this. And the reason we need better tools is because symptoms are nonspecific. So if we think about a rash, for example, which is a common sign or symptom of inflammation, that could be a fungal infection, that could be autoimmune, and how you treat that is really important and very different. So if you treat it with steroids as if it's autoimmune and it was really a fungal infection, that could lead to really bad outcomes. And treatment's urgent and it needs to be correct. So our solution to this is to look at cell-free RNA. So cell-free RNA are intact mRNA and non-coding RNA molecules that are circulating in blood plasma. So it's kind of where you least expect to find RNA. We think of RNA as a very fragile, brittle molecule, but we find that cell-free RNA is actually very stable in blood plasma, and it's repeatable and reproducible. And this is cell-free RNA is released from cells that are dying or activated, which if you think about it, are some of the most interesting during disease, the cells that are being most impacted or responding to the disease. And they're derived from cells in the blood and the tissues. When we look at cell-free RNA, we're finding RNA markers that are specific to the heart, to the lungs, to the gut, to the vasculature in the blood. So from a simple blood draw, you can get all this information in kind of a whole body molecular scan. And during my PhD here at Cornell in the Development Lab, I studied cell-free RNA in inflammatory conditions and developed machine learning algorithms to interpret these complicated profiles. And our platform at Romix is centered around this. So using a pretty small amount of blood, so we use 500 microliters to 1 ml of plasma, we can do deep cell-free RNA profiling through next generation sequencing and put it through these custom AI machine learning algorithms. Now there's two different outputs I can think of from our platform. One would be a kind of traditional diagnosis, a yes or no, does this patient have this condition or not? But I also think that cell-free RNA as a full body molecular scan is also incredibly powerful, where we can measure organ damage and immune activity that's distinct from biomarkers currently available. We've shown that cell-free RNA reflects immune activity in different ways than whole blood RNA, or different ways from proteins and complete blood cell counts and these sorts of things. So it's a whole new view of the immune system. And our engine, our analytics engine and our analysis is really unique because we are integrating lots of different data. So over the last decade, there's been an explosion of single-cell RNA-seq data that's been made available. This is RNA-seq data from individual cells from throughout the human body. So we know the RNA profile of these cells. And as I mentioned, cell-free RNA comes from the entire body. So we can take our RNA profile and compare it to these atlases to see where the RNA is coming from in the disease and what that cell was doing. We've also already processed over 3,000 clinical samples from a range of inflammatory conditions and other conditions as well. So anything from tuberculosis to pediatric autoimmune, immunotherapies, chronic fatigue syndrome, with a really well curated database of clinical samples to project new samples onto and better understand and compare. And these data sets, as well as with the most recent advances in AI and machine learning, I'd say over the past decade and what's coming out right now, allow for a deeper understanding of cell-free RNA than we've really ever had before. Our goal for this at Romix is to develop a universal blood test. I could see a blood test where a doctor orders the Romix cell-free RNA test, so we could provide diagnostic predictions, we can provide diagnostic scores, maybe immune cell activity that they currently don't have access to, as well as full body organ damage. Now that's a really ambitious goal, I recognize, and to get there, we need a lot of training data and we need a lot of clinical data. As we build our training data and as we train our machine learning algorithms, I'm interested in selecting that data in such a way so that we can develop in vitro diagnostic tests, laboratory developed tests, and function as a biomarker platform. So as we're building toward this universal blood test that gives a full body molecular scan, we're generating revenue and we're generating more traditional diagnostic tests along the way. So our beachhead markets are pediatric inflammation and cellular immunotherapies, which at first glance, you may say these are two very distinct fields of medicine, but really there's quite a bit of overlap. So they're both have systemic inflammation. Diagnosis directly guides treatment, and we're very careful at Romix to really only go after indications where the diagnosis is actionable, where it changes outcomes and therefore has some legitimacy. They're severely lacking molecular tools. So the pediatric space is very underserved in general. And there's really a need, and there's a lot of pediatric autoimmune and inflammatory conditions that are really hard to diagnose right now. The cellular immunotherapy space, on the other hand, is very exciting. It's moving very rapidly, and there's a natural lag between therapies developing and diagnostics and biomarkers and molecular tools to catch up with them. So for pediatric inflammation, we're interested in going a lot of different directions there. But our first product would be a Kawasaki disease diagnostic test. Kawasaki disease is the leading cause of acquired heart disease in children, and there is no diagnosis available right now. I'd be happy to go a little bit more in depth in that if anyone would like. But we're also interested in developing multi-disease diagnostic tests as well, more clinical decision support tools. For cellular immunotherapies, we have over 700 unpublished data sets in hematopoietic stem cell transplantation and CAR T cell therapy. And we're interested in functioning in a couple ways in this space. One would be developing diagnostics and very specific tests, potentially functioning as a companion diagnostic, as well as offering more insight into what these drugs and what these treatments are doing in patients during clinical studies and clinical trials, potentially acting as a molecular readout. So the technology is actually quite developed. I've been working on it for the past 5 years at Cornell in my PhD. We have 6 peer-reviewed publications and high-impact papers such as Nature, PNAS, Cell Reports Medicine, and others. We've filed 4 patents with Cornell CTL, which we have an exclusive option to license. We have over 3,000, as I mentioned, patient samples already processed, and have spent a significant amount on foundational research from grants and the like. The team right now, we're pretty lean still. It's just myself as the CEO and co-founder with Ewan Devlamek, who is my PhD advisor. Ewan has experience founding other biotech companies and is on the advisory board of other diagnostic companies and knows the space really well. And then Ewan and I are bringing excellent engineering while our collaborators are bringing really top-line excellent medicine. Jane Burns at UC San Diego is a pediatric inflammatory disease specialist. Jerome Ritz is a cell therapy innovator at Dana-Farber, and Charles Chu is a diagnostic leader, particularly in infectious diseases at UCSF. So we're bringing these 2 fields together in, I think, a really exciting way. So here's our roadmap. We're looking to leave the Ignite Fellowship in the spring. And that's when we're going to begin our fundraising. So we aren't fundraising right now, so I don't have a traditional ask. Any comments, connections, or feedback are all very welcome. But we're going to start to begin our large-scale studies on Kawasaki disease and graft versus host, move these, and as well as initiate pilot programs with pharmaceutical companies, translate and kind of develop these diagnostic tests and start interactions with the FDA in Q4 2026. But I think I'm at 8 minutes now, so that's what I have. I'd be happy to answer any questions, though. Thank you so much.
Thank you, Conor. Any questions for Conor? That was an excellent. Let's see if we have any questions here. Gus. See, how do we, Stephen, can we?
Yes, I just invited Gus. Yep.
Great.
Can everyone hear me?
Yep.
Terrific. Conor, I'm just curious about the competitive landscape.
Certainly. Let me move on here. I think there's a couple different ways to think about competitive landscape. In the cell-free space, we're kind of one of the first moving into the infectious and inflammation, and particularly cell-free RNA is really underdeveloped and expanding. In terms of bioanalytes, I think cell-free RNA provides a lot of information that's unavailable through whole blood RNA, cell-free DNA, protein, other liquid biopsies. There are other companies developing diagnostic tests and these sorts of things, but currently, for example, for Kawasaki disease, there is no diagnostic test available.
Got it.
Great. Sorry, go ahead.
One follow-up question. Of all the causes of inflammation, how many of them will show up in cell-free RNA? in the blood?
It's a really great question. So what we're looking at with cell-free is is the unique immune response and cell damage from inflammation. So for Kawasaki disease, we actually don't know what causes it, but we know that what we see in the cell-free RNA as diagnostic is the remodeling of the vasculature. So we're seeing the pathology that's unique to that disease in the cell-free RNA and the cell death and cell activation. So I think it's kind of universal in that sense. We're not picking up individual viruses though and measuring that, if that makes sense.
Okay, I got it. Thank you.
Thank you.
Thank you.
So Connor, one more thing. Do you have a website at all?
Not yet. We're working on that now. But no, we do not have a website up yet.
Great. And then another question, and then we'll move on to Tiffany St. Bernard next. But a person wrote a great presentation. They're curious about who your customers would be. Is there any reimbursement strategy associated with your product? And is this a 510K pathway? And then how do they reach you?
Yeah, absolutely. Really good questions. So it depends which product we're looking at and what kind of test we're going to develop. So we're not very interested in actually short-term putting boxes in hospitals. I'm interested in licensing this or developing our LDTs. But for IVDs, we're interested in licensing. For reimbursement, for example, Kawasaki disease, that's a longer path where we'd have to establish CPT codes, but we could start billing inpatient via DRGs. And then for the HSCT and the immunotherapies, it's a whole different path, and we would have to go through FDA. Yes, it would be 510K clearance, though, either way for LDT or in vitro diagnostics. Yeah. You can reach me on my email, cjl332@Cornell. I can put it in the chat, but I would love to chat more.
Great. And we will have a networking opportunity afterwards. Connor, thank you so much. Let's give you a big hand.